Hello, everyone. We'll be starting in 2 minutes. And in the meantime, those who have joined just now, it would be great if you could introduce yourself in the chat box, like your educational background, where you're coming from, and what are your research interests. Like we have Charles and Richard introducing themselves, just like that. So we'll be starting in 2 minutes. We're just waiting for the rest of the participants to join.
I have a question. Who all are available for this webinar? Like eligible, I mean.
Hello, Vansh. Thank you for that question. Yes, the high school training programs, as you can see on the screen. So the eligibility is for students who are in from 8th grade to 12th grade, as well as who are beginners and maybe undergraduates who would want to understand how bioinformatics and data science is applied in biomedical and other research, which what we will be covering in today's session, and letting everyone know that how this will be. Does that answer your question, Vansh?
All right, let's start today's webinar. Hello, everyone. My name is Sri Gauri, and I would like to welcome you all for today's webinar on OmicsLogic Bioinformatics High School Training Programs. And before proceeding further, let us discuss what the high school programs are all about. So to begin with, the high school summer programs provide students great opportunities to pursue new interests, improve existing skills, and brainstorm with like-minded students. And although bioinformatics and data science continues to redefine the way research is carried out, it has barely been incorporated into the high school education system. To address this disparity, Biotech has launched the OmicsLogic high school summer training programs, specifically designed to meet the needs and interests of high school students in the following areas of interest, that is biomedical data science using R and Python, genomics in virtual lab, and independent research project. So before we begin with today's webinar, let me briefly introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools and make bioinformatics more accessible. And over the past several years, we have had the privilege of working with a number of organizations, developing a training program for research-oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. The OmicsLogic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. Now, to mention the educational objectives, the OmicsLogic training follows a project-based learning approach using research-grade tools to analyze data from top peer-reviewed journals. OmicsLogic training has been completed by students, researchers, and faculty in 6 different specialization tracks, including oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. The OmicsLogic training has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine our local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. I would like to introduce you to the founders and team members of the company. Dr. Alfred Tauber comes from a medical background with an expertise in oncology, immunology, and biochemistry of inflammation. And one of his main projects is the establishment of the Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonard Brodsky is the director of Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a result of hard work by some of the people mentioned here on the right. Dr. Julia Pranav is an expert bioinformatician. Avi Tavisky leads the development of the T-Bioinfo platform. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in top peer-reviewed journals, and he has been developing collaborations with universities as a mentor and project lead for OmicsLogic training and research fellowship. And Dr. Raghavendran Lakshminarayan is an accomplished computation biologist with experience in large-scale analysis of multi-omics data and data science methods in precision oncology, protein or DNA structural bioinformatics. All right, so now let's highlight some of the current challenges and limitations and how OmicsLogic help resolve these with our training and research services. So to discuss some of the challenges that the field of bioinformatics is facing currently, to begin with, this field is constantly growing and developing new technology and software to improve processing time and power. The data sets that are analyzed are usually very large and take immense amount of computing power and memory that many personal computers do not have. Also, despite publicly available data, they're also usually complex to access, and coding knowledge is usually required to do so. Similarly, analysis pipelines and programs require coding knowledge that many biologists do not have. And in a university setting, students have the opportunity to learn the background information of designing and implementing a bioinformatics project, but do not receive practical hands-on experience with the full workflow due to the limitations mentioned previously. Similarly, faculties face challenges with being able to cover a diverse range of topics if they're specialized in a certain area of bioinformatics, or if they're unfamiliar with bioinformatics in general. For example, a professor whose research focuses on oncogene expression might not know about the metagenomics of seawater. To overcome these challenges, the OmicsLogic platform was developed. This program aims to standardize, organize, and systemize bioinformatics training in order to make these analyses usable for any biologist or clinicians. And some problems for standardization materials for these courses are usually developed in-house, taught sporadically, and are rarely updated. And the organization, because the challenge is to combine several different topics together, the flow and transitions are needed to be developed. Systematization, how can you know that someone is stuck and needs help or allows someone to run forward? OmicsLogic uses standardized modules and interactive sessions beginning with mastering terminology and concepts to implementing these concepts with real data sets in a project-based approach. Now, to discuss about the learning process, in order to create a clear path of skill development, OmicsLogic employs the Bloom's taxonomy levels of learning. Now, what this is, this approach starts with the basics. Students recall facts and terminology and are able to repeat and memorize the material. And with this base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new situations. The student will then be able to analyze data by drawing connections and evaluate when to use certain methods. This leads them to the highest level of learning, the ability to design and create original work. So how do OmicsLogic resources help advance bioinformatics teaching? OmicsLogic is a portal for practical and theoretical learning of bioinformatics, composed of courses designed to take you through the theoretical knowledge. And OmicsLogic takes a combination of training modules, data analysis tools, curated project data sets, and interactive sessions with mentors to give a student a clear path up the Bloom's taxonomy pyramid. No matter what the initial user's experience is regarding bioinformatics, OmicsLogic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic coding languages like R and Python, or beginning a data analysis project. And the users will be given directions to access the T-Bioinfo platform for bioinformatics processing and analysis of data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipeline. And different stages of analysis are performed in different sections on the multi-omics platform. This program comes with an access to the OmicsLogic courses, which includes courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, and epigenomics. Data science that includes bioinformatics using R and Python are also covered. And example projects of different fields which you could learn from and replicate for your research work, as displayed on the slide, including projects such as single-cell RNA-seq to TCGA, liver cancer risk. So first of all, let's begin by signing up on the OmicsLogic Learn portal. We'll share the link to the portal in the chat box, and I'll wait for a couple of moments so that users can sign up on the portal, and we'll resume after that. So once you have signed up on the portal, please put a yes in the chat box so that I know when to proceed. Sparsh has shared the link to the portal. So once you have signed up, please put a yes in the chat box. If you're facing any technical challenges, you can mention that, and our team will be here to help you sort through it. All right, Harish has signed up on the portal. Yes, Richard, it is. You can sign up on the portal so that you can browse through and see what are the various courses and example projects that we have to offer, so that you can align your interest accordingly. All right, so proceeding to the next part. So once you have signed up on the portal, this is what that you'll be seeing. So in an updated profile, a completed profile will have the following. That is a profile picture, as you can see on the slide, the full name with proper capitalization, link to your social media handles. That could be your Facebook, LinkedIn, Instagram, OrcID, or Twitter, and a brief bio about yourself that will consist of your educational background and mentioning your research interest. And under the various tabs that you can see, the mentors can view your progress, and the courses that you're currently doing will appear under the courses tab. And after completing a course, your certificates will appear under the certificates tab. All right, so now I'll just quickly take you through the signing up.
Do we go on the basic tab
so
or
in this case, since I've already signed up, I have the email ID and the password already saved. But if you are new to the platform, you can either use Google account or your Facebook or Twitter or GitHub account to log in, which will be easier for you. Or you can also sign up using your email ID, just like I did. So now I'm going to sign in to the portal, and I'll take you through the. All right, so if you click on the tab that says welcome back with your email ID, that will take you to your profile. And a completed profile will have, just like I said, your name with proper capitalization, a link to your social media handle. So in my case, I've linked the LinkedIn and my OrcID. And then I've mentioned my education background and my research interest. And the activity, that is under the activity tab, you can view your progress. And this is the place where the mentors will also be tracking your progress. And you have the courses tab, the projects tab, and the programs tab. So once you have logged in, you can browse through and check through the diverse range of courses and example projects that we have to offer. So to move on to the next part of the session, I'd like to invite Mr. Ilya Brodsky, who will give you a high overview level of the topics that will be covered during the program.
Thank you, Srigori. And just briefly answering some of the questions here in the chat. So first of all, if you have already signed up, you have a profile, there's no need to re-sign up. This is just to make sure that you have access, because we'll be referencing some of the things that we'll have on the portal. The second question is what to do after you sign up. You get redirected to a pricing page. There's no need to pay for anything right now. This is just to give you access. So just navigate away from the pricing page. And as we go through the program, it will be more clear how the different resources can be used. So In my part of the presentation, I will briefly talk about what the high-level topics are that we're going to cover and discuss how you might benefit from participating in the program, what might be the possible outcomes. I'll introduce you to some of the mentors that will be helping you along the way and show you some examples of students that have completed this program and what they have done with the information that they received. So as you all know, we live in the age of data. And specifically in biomedical research or in biology, there's a growing source of data. And one data in particular, called next generation sequencing data, is becoming kind of the foundation of a lot of the different research topics that are being done in biomedical research and agriculture and biotechnology, etc. So my background is actually not in bioinformatics, but I came into bioinformatics by working with Pine Biotech and the different collaborators that include multiple research universities, biomedical research projects and consortia. And I had the privilege of training and working alongside experts and students in many different institutions here in the US. So as a result of that, I've seen how it's really unpredictable to know who becomes an expert and who leaves and who kind of chooses bioinformatics as their topic. And for a lot of people, what I've discovered is it's not clear what bioinformatics actually is and how is it applied in a variety of domains. So my first part of today's session is to explain why bioinformatics and what is bioinformatics and how data in biomedical research is actually used to advance the application of insights from such analyses to advance precision medicine, biotechnology, agriculture, etc. So first of all, kind of a historical background. Since the 1950s, when the DNA was first discovered, the structure of DNA was clarified, we've seen how such information has led to new discoveries, starting from the sequencing, annotation, and discovery of the human genome and the genes that are responsible for different proteins, to technologies that make such data easy to produce and eventually to use in large-scale initiatives like Genomics England, the Broad Institute, different medical centers that actually use insights from genomic data to categorize, diagnose, and treat patients. Now, the basis of that is that when we understand the code and we understand what the code actually produces, so how nucleotide sequences form amino acid sequences that are the basis of proteins, we can start looking at variation of that DNA code that is responsible for some specific phenotype. Or in this case, in this example, you can see sickle cell anemia, actually a change in a particular amino acid leads to a change in a cell structure, right? And so that kind of an ability to detect changes has led to a number of very clear applications. So for example, drug metabolism, how different individuals metabolize different drugs, whether they will respond or not respond to treatment, identification of mutations in cancer cells, identifying genetic predisposition or disorders like sickle cell anemia, inherited mutations, rare genetic diseases, as well as drug resistance and different pathogens. And these examples are all in the context of biomedical research and medical application in the context of precision or personalized medicine. Because what happens in the medical context is today clinicians have to deal with such information to actually make a better description or characterization of disease for a patient. And one of the things that we now are starting to see is that you have these panels of specific genes, specific proteins, and you can now identify mutations that are responsible for very dangerous diseases like cancer. Now, as researchers continue to study these, they actually understand that some specific genes in the genome are responsible for the most serious effects. So, for example, tumor suppressor genes, oncogenes, DNA repair genes, they accumulate mutations that are most dangerous. One example is the TP53 mutation that is a gene, P53 protein, tumor protein P53 is encoded in that gene. And what that mutation does is it can actually alter the structure of that protein and make it not functional. And if it's not functional, it's a tumor suppressor gene. So then it does not suppress the tumor growth because it does not respond to the damage in the DNA and leads to accumulation of mutations. So finding such mutations can be critical for accurate and precise and effective cancer treatment. In cystic fibrosis, you have mutations that are responsible for the functioning of the CFTR channel, which is responsible for getting the right kind of chloride ions out of the cells. And if it's not functioning, it leads to accumulation of mucus and can be very dangerous. So knowing how to find these mutations and understand their function is clearly important. And not only is this important for individual mutations, this is also important for large-scale rearrangements like copy number variation, repeats that expand a particular region. And one way to find those variations is to perform genome-wide analyses that take thousands of samples for clear phenotypes and start understanding what are the statistically significant patterns that occur in a large proportion of the population. Such studies have led to discoveries, including tuberculosis drug resistance, which is a disease that is affecting millions of individuals around the world. If someone can monitor the participants and make sure that no one, thank you, if you could keep them unmuted, thank you. So drug resistance, not only to individual drugs, but multiple drugs is starting to become a huge problem. And we see the same in the world of viral pathogens. So for example, the Ebola outbreak and how that leads to tracking of transmission and vaccine design, but also the current pandemic of COVID-19. We hear about variants of concern all the time. So all of these project examples, all of these examples in precision medicine, in population health, genetic predisposition to disease, all of these have been transformed by the availability of these different types of sequencing techniques. So in the program, what we will try to do is introduce you to how such data is generated. What is the process of acquiring actual digital data from a biological sample? And then in that data set, how do we work with different algorithms, computational algorithms, to understand mutations that are pathogenic, neutral, that are maybe not very bad or bad? And how do we determine the effect of these mutations in the genome? So how do we actually categorize them? How do we understand their impact on the phenotype? And what are some examples where you can find the clear association between individual mutations and the phenotype? And so that is really what bioinformatics is all about. So bioinformatics is about taking information or data, taking methods of computational analysis, statistical methods, machine learning, analytical methods, and applying them to biological data sets, applying them to data sets where we have biological samples, conditions, clinical conditions, et cetera. And so the program is going to tie together these three different domains in a way that you can quickly see the application of bioinformatics to a number of different domains. So the OmicsLogic programs are going to help you not only understand how these things work, but also practice and apply them. And so in the context of this program, these resources that were already mentioned by Sri Gauri are going to be critical to getting you from maybe not knowing a lot about bioinformatics to getting an understanding of what bioinformatics is and how it applies, to eventually starting to use it, maybe in your own project or a group. And so here, we're going to try to cover a variety of different types of examples or projects that are case studies that you can use to really understand and develop a sense of you personally discovering something on your own using some of these tools. Now, I imagine that most of you here have understanding of what bioinformatics is, maybe not a lot of experience, and that is really who the program is for. Now, I want to specifically highlight two two resources on the OmicsLogic portal where you can get started. So even if you're here for the first time, now that you have registered, you can go to the OmicsLogic portal and find two specific courses. One is called Introduction to Bioinformatics, and another one is called Bytes and Molecules. So Sparsh or Srigori, somebody maybe can, someone can maybe post the links. So if you have an account and you've signed up, again, the sign up is free. Those are two introductory courses that will get you started. They will get you into this context. They will explain at a high level the different applications, introduce you to our community, the way that things are organized, and you will really get a good sense of why bioinformatics is it for me and what can I do with it. Okay, thank you, Tash. So Introduction to Bioinformatics is that first course, and the second course that I wanted to mention is called Bytes and Molecules. So again, if you search here, Bytes and Molecules, it is course 2. And this course is different from the Introduction to Bioinformatics because it is a little bit more detailed and actually introduces a lot of the concepts that are going to be useful for what we do in this program. And that includes terminology, or what are these terms like SNPs, variants, gene expression, profiles, etc. It will give you a sense of the relationship between code and structure and how things work in the cell. And then it will introduce you to specific types of data that could be acquired from next generation sequencing, so genomics, transcriptomics, and metagenomics, and talk a little bit about maybe things that you haven't thought of how bioinformatics is actually used to study precision medicine or astronaut health in space. Now, also a big component of bioinformatics, as I mentioned, is the application of data analysis techniques and tools to large data sets. And coding is a critical component. So as you've seen, one of the critical elements is to understand how and why, and then have the practical skills. And so the second option that was mentioned in the beginning, so the first one was genomics in the virtual lab. The second one was coding or biomedical data analysis in R and Python. So in this course, you will already get an introduction to coding in R, which is a popular method of analyzing different types of biological data, where you will have access to different examples of just getting to know what coding is, why do we need coding, and how to read, produce, to load the data, things like that. So that's going to be very useful because one of the practical examples here is the ability to actually run code and practice fixing the code that is given to you without having to take too much time of yours to actually learn everything from scratch. So we're not going to learn things like hello world. We're going to jump into how to load data. And this data is going to be biological data, or the type of data that you are expected to use in the domain of bioinformatics. And once we get into this, you will see that you will start to appreciate syntax. You will know how to load and produce simple plots that are informative. You to interpret these plots. And then as it becomes more and more complex, so here you will find assignments, those will introduce you to more and more complex methods of analysis, including analysis of complex data, analysis using complex methods like machine learning and deep learning, and also ability to integrate multiple sources of information to actually answer real research questions. So along the way, as you go through this coursework, you will realize that a lot of this coursework is sourced from experts in the field. And experts in the field are people that have a long history of maybe doing research or using such skills in the domain of biotechnology and pharma. And getting to know these people, you will realize that they all have a rich history and a path of how they got to where they are today. So to briefly introduce some of these people, I want to highlight a couple examples. And those examples are going to demonstrate kind of the quality of the people that you will be exposed to. So let me just open that video real quick. And what I will play is a short video about a couple of people that have contributed to the development of the coursework that we will be sampling. And hopefully that will clarify who these people are. And then I will pass it on to Dr. Raghav, who will speak from his personal experience about his mentorship experience and what he does with that now. Okay, can you hear my?
Let me know if you can hear this.
It's just been a great meeting. Talks and posters. I'm very excited. And so what's interesting though is as I was preparing this talk, I realized that today is 25 years to the day since I started my postdoc. And so I had just finished my PhD work under the direction of Dr. Bruce Rowe at the University of Oklahoma, where we were sequencing regions of human chromosome 22 as part of the Human Genome Project. And on April 1st, 1997, I began my postdoctoral study at the NIH under the direction of Dr. Francis Collins. Now, I can tell you, if you've never started a new job on April Fool's Day, it is quite the experience. And so as the new green postdoc, I was immediately the April Fool and the easy target of many practical jokes in my new lab. But what I didn't realize at that point was that this was the beginning of a translational career built on genomics and its application to human health. And so I was fortunate to not only contribute sequencing data to the Human Genome Project, but to also be in the epicenter of human genomic research in the US upon completion of the Human Genome Reference Sequence in 2003. So at that time, I was infused with optimism about what we could do to understand human health and disease, how we could further understand what makes each of us alike, but also what makes each of us different. So how are we different in our responses to environmental stimuli, to pharmaceutical drugs, to chemotherapy? And since this day in 1997, I've studied cancer and performed large-scale transcriptomic analyses of several tumor and cancer syndromes. I've worked in the drug development and pharmaceutical industry. And then now here we are 25 years later, I'm translating what I've learned in each of these varied research environments to supporting patient management in our CAP-accredited clinical precision medicine laboratory.
So really, as you can see, the people who have worked in this domain for many decades and have now been able to apply what they have learned and studied to actual practical applications are the types of people that you will meet throughout this program virtually and be able to work through problems that represent the type of work that they do to really understand the impact of bioinformatics in the domains of precision medicine, basic and translational research. And so with that, I would like to introduce you to a mentor that will help you throughout these projects. And his name is Dr. Raghavendran Lakshminarayan. He will be one of the mentors that will guide you through the project. So Dr. Raghav, over to you.
Okay, thank you very much, Eli. I think something's wrong with my video. Let me fix it.
Kind of an effect that you're using.
No, I don't know.
Change your video background preference. So
yeah, I think that should work. Yes, perfect. Thank you very much, Eli. Let me find ways to share my screen. So can you see my screen and am I audible?
Yes, both, yes.
Perfect, thank you very much. I am Raghavendran Lakshminarayanan. I'm a research consultant and bioinformatics mentor with Pine Biotech. It's wonderful to see you all, and welcome to this session. I'm trained in bioinformatics and I hold a computational biology PhD degree from Max Planck Institute, Germany. So my research interest and focus is on the application of variety of data science techniques such as machine learning approaches, deep learning approaches towards the translational studies of cancer biology, towards applying to plant genetics and neurological disorder, etc. So my role in this company includes mentoring a variety of students like you, students who are from high school, students who are from bachelor's, students who are doing masters, and all the way up to students who are pursuing PhDs. So please let me introduce you about my background and how I started in the field of bioinformatics research. So the impact of Human Genome Project in a variety of biological research field is very well known and documented. So in fact, this very discussion that we are having and the meeting is a result of this extraordinary project. So this project affected my career as well. So when the findings of Human Genome Research Project was announced and its promise inspired me to pursue a career in biology from jumping fields from being a physicist. So I completed my master's in biophysics and bioinformatics, being a undergraduate in physics, and followed by which I joined Max Planck Institute in Germany to work on my PhD thesis. So after that, my two postdoctoral research tenures from India, from Institute of Bioinformatics and Applied Biotechnology, Bengaluru, and Jawaharlal Nehru University, this is where it was instrumental in helping me develop a deeper understanding in the area of bioinformatics research applications to a variety of different data sets. So for the past 8 months, I am part of this wonderful company, which gives me opportunity to share my learning to inspiring students like you. So over the course of my career or over the course of my tenure, I have been involved in projects from a variety of different backgrounds. That includes protein structural biology, that includes agriculture, that includes multi-omics data analysis like genomics, transcriptomics, epigenomics, data, etc. And these omics data are acquired from a variety of different sources like model systems, like from patient resources, and like from agricultural sources, and etc. So this variety of projects and this wide spectrum has been immensely helpful for me to tending to students with a wide range of interest and in pursuing research training with us. So to give you an idea of my research interest and background, let me share one of the interesting projects that I was involved in during my career. So this is a project on precision oncology. And to introduce you to precision oncology, first let me tell that most of you would have already known, oncology is a study of cancer biology. And we know that cancer is the leading cause of disease deaths worldwide. So for such a dreadful disease and for disease with such an impact, systematic carrier treatments have relied on drugs which are chemotherapeutic drugs, and these drugs are marginally more toxic to the cancer cells than normal cells. I mean, such an approach is not very ideal, and it gives us or results in a huge problem because on an average, only 25% of the cancer patients respond positively to these cancer drugs. But in comparison, over 80% of the patients respond to popular painkillers like aspirin that you get over the counter. So one of the prominent solutions to this problem is precision oncology. So precision oncological therapy is a strategy where the treatment is actually designed by targeting the molecular characteristics of the patient's tumor. So hence, the treatment that is given to the patients are personalized to those patients who are likely to respond to specific therapies and therapeutic approaches. So the way that we have approached, or the way that I have addressed this precision oncological problem issue, is to implement data-driven approaches to include multi-omics data for understanding and for predicting patients' response to a variety of cancer treatments. So how do I do this? The tumor cells that form cancer in patients are collected during biopsy and are then subjected to next generation sequencing techniques, and the information about gene expression, the information about mutation, and the information about epigenetic variation that characterizes the gene expression were extracted and they were recorded. This data, or these omics data, molecularly characterizes the tumor. So these are different for different types of tumor and different subtypes of tumor. And then these tumor samples are then grafted or then administered several cancer drugs, and their response is studied by measuring their volume. So this data records the tumor's reaction and response to different treatments. For some treatments, the tumor will diminish in volume, and for some of the treatments, it will not respond positively. So these responses and the molecular characterized data were then integrated, and various machine learning algorithms and deep learning algorithms were applied to understand how they are related. So from this relationship, we were able to predict which patient or which cancer from which patients were able to positively or will be able to respond positively to the treatment that they are subjected to, or from a list of multiple treatments. So this is how we approach understanding cancer biology from multi-omics data driven approaches. So projects like these and various other projects and example projects, methodologies, and resources are compiled and explained expertly in the Learn OmicsLogic platform. So while you are enrolled in specific programs that addresses your interest, you are also welcome to explore various such resources and example projects and expand your horizons in the field of bioinformatics data analysis. So as I have already mentioned, my main involvement in this is with the students who are enrolled with research fellowship program. This research fellowship program is designed to address key challenges that every student will face while subjecting or while opting for a bioinformatics research. So if you are interested in extending your enrollment for a research fellowship program after the completion of your specific program, then you would also be able to leverage all the resources that are developed to overcome these key challenges, like time involved in learning and testing out a lot of different strategies, and time and access to bioinformatics expertise and access to big data infrastructure. So who will benefit from a research fellowship program like this, and what will the students like you take away from these research fellowship program? So for students who are new to bioinformatics and who does not have an early exposure to bioinformatics, this gives you an excellent opportunity to develop an exposure to the methods, to the logic behind these methods and analysis, and to practice your knowledge that we have gained throughout this program into a research project that interests you, or research project that will help you in your career. And for students who have some exposure to bioinformatics and who already know a little bit of bioinformatics, you will gain expert feedback and project evaluation that will take your project from superficial investigation to a full-fledged research project that fulfills many prominent criteria for being a successful publication. So you will also be interacting with mentors like me and Dr. Mohit and Ilya, and understand where and how to find complex data sets that is going to help you design a very specific bioinformatics project. And this project, as I have already mentioned, can be developed or can be designed with an aim to obtain a research publication by the end of your tenure. So with that, I will conclude my presentation, and I look forward to interact with all of you during the program and see most of you for the research fellowship program. And what happened? Yes. So now I will pass the stage to Dr. Mohit Majumdar, who will talk about the highlights and curriculum of this summer program. Over to you, Dr. Mohan.
Thank you, Dr. Raghav, and hello, everyone. My name is Dr. Mohit, and I'm going to share my screen with you all so that we can go through the curriculums that we have designed for this summer and all the opportunities that are available. So I'll start from this page, the page that has been shared with you, and many of you might have visited this page. So this is the page that describes the high school summer training program in much more detail. And you'll find a lot of resources here, a lot of information that will help you understand more about the design and more about the curriculum, more about the topics, and more about the schedule of the programs, upcoming sessions, and all of that. So let's begin with an overview, as you've already learned that what are the major topics and how we are going to do this. And if during this presentation, if you have any question, please feel free to let me know by putting it in the chat or unmuting yourself. So as we are having the first webinar here and we are discussing about the program, we are going to offer 3 different types of training, which will be available for all of you interested to participate in. So these 3 types of trainings will include different types of topics, different types of outcome. So let me explain that like very briefly. So we have listed out some of the advantages and how this training is going to be, including what all topics and how the system works. You already created a login on Learn, so that will help you to orient yourself and get comfortable with the resources that we'll be sharing. So you have an option, as I was saying that we are going to invite students to join these programs which are available. So how do we learn about what will be the topics, and which dates they will be covered, and what will be the resources which will be put together for those sessions. So let's take a look at by clicking these specific programs. So we have listed out here like 4 programs, from learning about genomics and working with an expert to do some genomic research utilizing different resources, then to learn about biomedical data science using coding by applying R and Python, and then to be able to do your own research by participating in an independent research project. So from understanding very beginner level courses, by understanding and going through the mentor and the support team, you'll be able to develop that understanding of how to utilize big data and how to utilize these different applications that you heard of. So I think in this meeting and during the call, you might have heard many terms which were new to you. If that is true, please let me know. If put a yes in the chat box. So for you to learn about those new keywords and new terminologies, we have prepared a lot of resources that will definitely going to help you go through them strategically. So let's first go through the first program. Genomics in the Virtual Lab, very special program where we have an excellent mentor who will take you, Professor Sona Vasudevan from Georgetown University, a very prestigious university in the US, and along with our mentors, which will start from 18th July, and it will be available for you to enroll for 1.5 months, that is 45 days training, or a 60-day training, or a 90-day training, based on your availability and the time that you can allocate for such training. So that's the timeline for the program to start. You will also find like a overview, and you can see that how the previous year student presented, and we will be going through some of these outcomes of the program, because it's important to understand that what happens after the program, what are the things that we are going to learn, and if there is some projects that we're going to work on. So we will be talking about that, but before that, let me take you through the schedule first, and a little bit about how do you join the program. So 18th of July, we have the first session, which is Introduction to Bioinformatics, very basic, which involves understanding of omics technologies, role of big data in biomedicines, and then different repositories, resources, and tools, how to access the Learn OmicsLogic and its projects and courses, how to access the Tauber Bioinformatics research platform, and start, using some of the pipelines. So it's very exciting. But yeah, we will start with a very basic overview of how that all of this is going to work for you. So that is the first session, which is scheduled for July 18th, which will be a live session. Then we have these different sessions which will be asynchronously available, which means that you will be getting the recording and you will be getting all the resources and whatever you need for to learn the topics that are going to cover on July 22nd, and an opportunity always to talk with us, some of the mentors who will be available for you to help understand some of the concepts, some of the topics, some of the problems that you face. So there will be a team supporting all of this. Then we have the next session, which is more about understanding how we can do comparative sequence analysis. How can we correlate it with the evolutionary analysis? So we will learn about sequences. From sequences, we will understand how sequences fold into three-dimensional structure, and how do we analyze the structure from sequence? How do we analyze those three-dimensionally modeled structure in our laptops, in our systems, and understand about different types of mutations maybe that causes the important or changes in the body, right? So we will be learning about all of those, from sequence to structure and then to function. And to understand function, we will take some of the case studies and we will review some of the diseases types that we have described and discussed in this webinar previously. So I'll not go into the detail, but one of the activity that is planned, and which will be one of the landmarks or highlights of this program, is that you will have an opportunity after the first 5 sessions to work on a small project and showcase. It's like a more of a review to find out some of the insights from what you have learned and apply to some of the projects that we are letting you know, or you can also give your own proposal. So this weekend activity will include preparing a couple of slides and sharing with us and discussing if you can work on this project, if this project is feasible, or if this research is feasible. So that would be about finding a research question. Followed by that, there will be specialized sessions which will cover very important topics for research, and very important topics for learning, which is to how do you do literature review and how do you design a project, and then what are the different elements that will be designed inside a project. So on August 8th, we have that session where we'll be sharing you how to design a bioinformatics research project, how to do literature review and all of that. And then we will dive into some sort of specializations, which is like virology here. The virology, you all have witnessed the COVID pandemic, and we will be learning about different aspects of virology and how bioinformatics is applied in virology. And it will involve understanding of how do you apply some basic R programming by understanding and utilizing some sequences, and then playing it in the code playground, and then doing it in the code playground as you have seen. So followed by that, we have some sessions on microbial communities where we will learn about metagenomics, because it's very important now to correlate between these different technologies and the data that they produce. So it actually helps us in developing a very good research project. So all of these sessions are kind of designed to help you develop that understanding, that ideas will automatically come when you understand that how they are applied in different case studies, and how they are applied in terms of doing it by doing it yourself on T-Bioinfo platform using the hands-on analysis. And then we have some case studies which involves vaccine, antibody, and antiviral drugs, and then analysis of genome on NCBI viruses, where we will be exploring the public repositories, because a lot of data, and huge amount of data are being deposited every day. And we all think that there is an opportunity to investigate further into those data sets, big data sets, which is usually very difficult to do, but otherwise it will be much easier through the program and through the resources that we'll be sharing. And that's an opportunity for all of you to take. Now, after this the second half of the program, where you will understand and go through all of these public repositories, tools to use, and technologies to apply, you will have an opportunity to do a project presentation where you come up with your own idea, and we will be there will be judges from academia and industry, and we have an amazing panel of judges who are from top universities, leaders in their field, can give you really good feedback about how the project is shaping up. So that's how the program is planned. And after 26th of August till the end of your program, you will have an opportunity to discuss with the mentors and the team and work on your research project and research proposal. And if you have a good research project proposal, then there's an opportunity for you to join the research fellowship and work on an independent research project that I'll come next. But before that, let me take you to the registration process. So if you have already created your login here, what all you have to do is just go to this page, and then go to this section where you can choose a subscription. Click the subscription button where you want to choose the subscription, and then go through these simple steps and complete your registration. If you're from low income country group, then you are eligible for scholarship, and you'll be able all you have to do is that write an email to us suggesting the same, and we will be able to guide you with the scholarship process. Now, going back to the second program, we have these different opportunities that we have been discussing. So please feel free to ask any questions related to the program that we are discussing. The next program is focused more on data science, more on doing coding, more on learning how to apply data science techniques into using biomedical or biological data. So maybe some of you are more interested in data science, some of you are more interested in investigating or doing biological research. So it is based on your interest. You can join and choose between these programs. So now the next program that I wanted to show you is the data science program, which is going to start very soon on 4th July, where we will be going through the basics of programming for everyone to get to know about get on the same page, perhaps, because we have students coming from different backgrounds, and many of those students do not have a background of programming at all. So we have these very beginner level sessions to help students learn from the mentors about these how to get started, and then installing and going through the very basics like vectors, matrices, libraries, and then start utilizing. Once you start utilizing, you start working with sequences, you learn about how to process data, and we start visualizing in R, and then you go into different type of advanced visualization, and you learn about machine learning approaches, and you understand that how can you use dimensionality reduction processes, or dimensionality reduction, or unsupervised machine learning processes that can actually help you to find needle from the haystack. Literally, from like large amount of data, you'll be able to find meaningful information using some of these techniques, and then you will be learning about more statistical and advanced techniques that will help you work on a project which is more related to data analysis, application of data science, and machine learning and all of that. So we have detailed session covering all of these important topics, and then we have a specialization that you can opt for. You can either do both R or Python, or you can choose one of these. It's up to you, but we will have sessions for both R and Python. So first we will cover R, and then we will cover Python, so that you can understand, compare, and take advantage of both of these very, very powerful programming tools that helps biologists, that helps almost everyone in today's era of data to gain meaningful insights and show that in a proper presentation and get recognition. So that's about the program that we are discussing. And I think we have also like a different dates, which is to cover which will be covered in 45 in 1 month's time. So you'll have 1 month time where these sessions are going to happen bi-weekly. And then you will have an opportunity to discuss with the mentors and learn about different sections. And you will also have the asynchronous coursework associated with each of these sessions. So those coursework are available on Learn OmicsLogic, and you will be able to get hold of that once you register, and also once you start the program. So I'll just log out and I'll just check because I'm already a part of this program, and it's not showing me the registration part. So I'll just show you the registration part. So as you can see, the asynchronous material is also included here. You can click on these links and go to the asynchronous material and check what all we will be covering in the session. Along with that, we have recorded videos that we will be sharing with you so that you can do it on your own if you don't get time. And we will also have flexibility in terms of scheduling meetings. If you are in US, we will have a US team supporting this and have a time convenient for all of us to have the meeting. So similarly, you can choose between the different subscription levels here, and also apply for scholarships if you're from low income country and have financial problems. Now, just the last part of the presentation or the discussion, where we are going to discuss, once you have learned all of this, once you have mastered one of these techniques, once you have practiced on example data sets, the next thing that you would want to do is to do it on a new problem. That is something that is like a contribution to the research and the scientific community, because these findings will be new and they are eligible based on their novelty for publications or for other poster presentations and such recognition. That program is the research fellowship program. And now like taking you through this different types of opportunities. So once you have that, like a research project, a research proposal, there's an opportunity to present that and publish that, and all of those connect with the research community using the Omics Research Symposium, which is scheduled to be happening in August. So all of this will make sense to get a really good outcome from this summer. If you're interested in exploring some of these topics, doing some research and using the skill sets for your future. So some of these key topics are also mentioned here, and this all has been kind of covered. We have excellent mentors who will be supporting the program. You can watch the video from one of these basic training sessions, initial sessions, for everyone to be on the same page. And then this research fellowship program or independent research project is the highlight, because here you will be able to see that there are videos from the students who completed these research trainings and come up with the wonderful outcomes. So let me explain or discuss a couple of case studies here. I want to tell you all about Hannah, one of the champions of the program. So Hannah worked on type 1 diabetes. Type 1 diabetes is important to Hannah and her family. So determining to find the root cause, she turned to bioinformatics and joined our collaborative OmicsLogic high school training program, where she used big data analysis tool sets that combines biology, computer science, and statistics. So let's take a look at her project. And if you're interested, then I can play the video. So let's play the video.
Hello, my name is Hannah Davis from Haytah High School, and my project is entitled An In-Depth Look into Identifying the Differentiation Pathway and Cell Type of the Dual Receptor Expressing Lymphocytes in Type 1 Diabetes. bit of background information about my subject. Type 1 diabetes is an autoimmune disease in which the body attacks its own islets of beta cells within the pancreas. The most common treatment for type 1 diabetes is insulin injection, and the average sufferer spends between 6,000 to 7,000 yearly on insulin alone. Diabetes is a disease of the immune system, which is the body's first defense against intruders known as pathogens. The immune system can be divided into the passive, active, and innate responses. Within these responses, you're going to have cells known as lymphocytes that officiate immune response. However, when the immune system does not work as planned, the immune cells target the body's own functioning cells as faulty cells and marks them for destruction. One new cell found in relation to type 1 diabetes is the DE cell. The dual receptor expressing lymphocyte is named because it displays both a T cell receptor and a B cell receptor. And according to the article in which the cell was originally published, these cells are shown to mimic insulin peptides to trigger diabetes-specific helper T cells. And although it could be supported that the DE cells are driving type 1 diabetes, this was not explicitly stated in the article that these cells were originally published, and it could not be adequately concluded by other scientists researching this topic. My project, in order to interpret the data, I'll be using bioinformatics. And within bioinformatics, I'll be using RNA-seq analysis, and within that, I'll be using differential gene expression analysis. So the purpose of my research is to provide as much information as I can about the DE cells to hopefully provide insight to see if these cells actually cause type 1 diabetes and what their role is in causing the disease. Furthermore, identifying the cell type and differentiation pathway of these cells could provide insight into other diseases, as well as prevention and reversion methods for type 1 diabetes itself, which currently do not exist. So the materials that I used, first I used the NCBI SRN table from this bio project right here. And I chose this data set because it's the original data set the DE cell was originally published. And the name of the article is a public BCR present in a unique dual receptor expressing lymphocyte from type 1 diabetes patients encodes a potent T cell autoantigen if you're looking to find that on NCBI. And I also used a computer with the capabilities of running my bioinformatics platform and used the Tauber Bioinformatics T-Bioinfo platform for RNA-seq analysis provided to me from Science Coach. I also used Microsoft Excel to interpret my data. Okay, for my procedure, first I downloaded the SRN table from the bio project in question. Then I input the files into my platform, selected 2 cell types with 3 samples each to compare at a time. Then I ran a pipeline. Specifically, I chose the top hat, cuff merge, cuff diff pipeline for differential gene expression analysis because it output a normalized expression count, which would be better indicative of the expression of the set as a whole since I only used 3 samples. And then I downloaded the gene expression output files and drew conclusions based on these outputs. So here's an example of some of the graphs that I made. This is a DE cell's top 50 expressed genes. Specifically within this, I'd like to point out the expression of this natural killer granzyme protein, as well as this immunoglobulin heavy protein being in the top 50, as well as a couple of these mitochondrial RNA genes being expressed, which would indicate a high proliferation rate of this cell. Here's the natural killer gene signature being expressed at very high rates by the DE cell, but not so much by the T and B cell. And to note that the NK cell and T cell are very similar in nature. However, the DE cell is showing that it is closer to a natural killer cell rather than a T cell by expressing more of these genes. Specifically, I'd like to point out perforin and some of these chemokines that are being excreted. This is the B cell gene signature being expressed by both the DE cell and the B cell alike. Here you can see that the DE cell is expressing cytokines as well as the TCR. So in conclusion, the DE cell is going to possess lineage markers indicative of a CD8 NKT cell, which suggests that the DE cell has B cell features because of its germline encoded BCR. And I was able to conclude this because in the original article, it stated that the BCR on the DE cell is of germline configuration. And this supports the idea that the DE cell is a dual purpose cell. So this leads to the differentiation pathway. So you can see that the DE cell is going to follow a relatively standard differentiation pathway of a natural killer cell. However, within TCR activation, it's going to be activated by self-antigen binding, and it's going to stop at the naive NKT cell and not develop beyond that. And after that, it's going to travel back to the bone marrow, where its BCR is going to become activated again by self-antigen binding, is what I hypothesized. And I concluded all this based on the expression of different cell markers being expressed, as well as the different TCR and BCR genes being expressed. So this is the proposed cell function model. Here you can see the DE cell, and it's going to identify this beta cell islet through.
So I'll pause it here, but what I wanted for all of us to see is that how you can use data which comes out from the analysis, which can be done through easy steps and then correlated with the biology. And this can be done within a very short period of time. Once you learn about how to use the platform, how to understand from the coursework and run some examples, and know how to do this by doing it yourself, practicing. So I mean, this led to a lot of accomplishments for Henna, and you can read a lot about it from here, numerous awards, so many that it will be difficult for me to read out. So please. So next, one of another very gifted and student, Andres, I want to discuss, who won the prestigious Potemkin Award. So Andres is from high school at Mexico, in one of the high schools in Mexico, and he was mentored by all of us here and worked on a project which was on in silico transcriptomics analysis of microglial cells that led to a really innovative and groundbreaking work in one of the in some of these study for brain in neurodegenerative diseases that led Andres to win the award, and also get an award, 4th award in biomedical and health science category at one of the very prestigious pharma company. So maybe we can quickly take a look at his project initial part where he's describing the problem, so that you understand that what different students can come up with their different ideas and can easily work by finding the data set and then applying some of these algorithms working with the team.
Hello, my name is Andres Modano Sosa, and I am a high school student from Mexico City. I'm currently an OmicsLogic Bioinformatics research fellow, and I am truly passionate about biochemistry, systems biology, and neuroscience. I am particularly interested in the biological mechanisms that underlie aging and neurodegenerative diseases. Over the last 3 months, I've been working on my research project together with my mentor, Dr. Harpreet Kaur. My research project seeks to evaluate the role of microglial cells in the pathogenesis and progression of Alzheimer's disease. Alzheimer's disease is a progressive and irreversible neurodegenerative disorder that results in the development of cognitive deficits and clinical dementia. Alzheimer's disease is characterized by the development of 2 pathological hallmarks: senile plaques, which arise due to the aggregation of the amyloid beta peptide, and neurofibrillary tangles, which arise due to the hyperphosphorylation of the tau protein. Although these 2 hallmarks are widely recognized as the 2 core pathologies of Alzheimer's disease, the mechanisms by which they are linked remain largely unknown. Over the last decade, the role of neuroinflammation and in the pathogenesis and progression of Alzheimer's disease has become more and more evident. Patients that suffer from Alzheimer's disease show an elevated level of inflammation in their brains. This inflammatory response in the brain is mediated by microglial cells, the brain's resident macrophages. Scientists have shown that the immune activation of microglial cells leads to the secretion of pro-inflammatory cytokines, and this inflammatory response has been shown to exacerbate both amyloid and tau pathologies. Therefore, many scientists have begun to appreciate neuroinflammation as the 3rd core pathology of Alzheimer's disease, as it may provide a link between the accumulation of senile plaques and the later development of neurofibrillary tangles. The purpose of this research project was to evaluate how the transcriptome of microglial cells varies both between physiological and pathological conditions, as well as how it changes throughout aging. For my analysis, I used the data from the following study: transcriptomic profiling of microglia and astrocytes throughout aging from Pan et al. This study evaluated the transcriptome of microglia and astrocytes from both wild-type and APPPS1 mice. These are double transgenic mice that exhibit a remarkable elevation of amyloid beta aggregation. And the data that this study provides yields valuable information regarding microglial activation in response to aging and amyloid beta pathologies. I performed an RNA-seq pipeline to obtain the expression levels in raw read counts and outputs. So to obtain a gene expression table with the expression values for the respective genes. And then using those raw read count values, we performed an exploratory principal component analysis to see how these samples clustered. And so here we can see that there isn't really clear clustering between wild type and AD samples. But here we can see that when we label samples in terms of age, we can see that 9 and 12-month-old samples cluster apart from 2, 4, and 6-month-old samples. And based on this observation, we defined 2 groups in terms of age. So with the young samples, which are 2, 4, and 6-month-old samples, and the older samples, which are 9 and 12-month-old samples. And yes, so here we can see that the young and the young wild type and the young AD samples cluster together, whereas the old wild type and the old AD samples cluster apart in the lower region of the plot. Then we perform a differential expression analysis using DSEC2. We perform these 4 different differential expression analyses. So first we compare the transcriptome of young AD and old AD samples. So this allows us to see how the transcriptome of microglial cells vary throughout pathological aging. And we did the same by comparing young wild type and old wild type samples to see how the transcriptome varied throughout normal non-pathological aging. And then we compared young wild type and young AD samples, as well as old wild type and old AD samples to see the phenotypic differences at age matched groups. And then here we quantified the number of differentially expressed genes produced by each differential expression analysis. And we can see that the greatest number of differentially expressed genes were produced here between the young AD and old AD, meaning that there is a greater phenotypic divergence throughout pathological aging when compared to normal non-pathological aging. And here we can see that there was a rather small number of differentially expressed genes between the young samples, which is telling us that there isn't really a phenotype, there aren't really great phenotypic differences between samples, and this supports the age-related nature of Alzheimer's disease, as phenotypic differences between healthy and diseased models arise throughout aging, and they are not present at a young age. Here we performed these volcano plots, and here we can see that there is a greater phenotypic divergence between the old wild type and the old AD samples and the young AD versus old AD samples. Here we can see that there isn't really a great number of differentially expressed genes between the young wild type and young AD samples. And here we can see that although there is a phenotypic divergence, the number of differentially expressed genes is not as large in here as in the young AD versus old AD samples. Then we performed principal component analysis using only the expression values from the differentially expressed genes. We considered differentially expressed genes, so those that have a p-value smaller than 0.05 and a log-to-fold change value either greater than 1.5 or smaller than minus 1.5. And then based on this, we performed the different clustering analyses, the principal component analysis, I'm sorry. And so here we can see that there is clustering between young AD and old AD samples. Here we can see that there is clustering between old wild type and old AD samples. And here, interestingly, we could see that there was also, here there appeared 3 clusters. So here we see one with 2 and 4-month-old samples. Here we see another one with 9 and 12-month-old samples. And here we see one with 6-month-old samples only, which was rather interesting. And here we can see that between the young wild type and young AD samples, they are all like skewing towards one of the axes. And so there isn't really a clear clustering or a difference between the experimental groups.
All right. So here, what I wanted all of you to understand is that in-depth understanding of the data that you're using, and applying different type of visualization methods, as well as, based on the analysis, refining your data further, can give you very good patterns that are can differentiate and show clustering, differentiation in clustering, and you can group them, and that would give you a clear pattern. So all of this pattern recognition, understanding, finding, and doing big data analysis is sort of possible within a short period of time in this summer through these training programs. So I'll just go back to the page that we were at. So this is the research fellowship program, the last program I wanted to explain to all of you. The research fellowship program is something that helps develop independent research projects. So if you think you're ready to work on an independent research project, you're more than welcome to join the research fellowship program, which is available for you to join anytime now. It is available since June 15th, and there's an opportunity to work in a 45 days project, 60 days project, or a 90 days project. The dates are not so much relevant here, because you can join anytime and you can start working on your project. So similarly, we have the pricing, and you can check out that and decide which subscription you would want and which program that you would want to enroll to. So that's the part of my presentation, and I hope all of you have enjoyed this webinar and learned a lot. So if there are any questions, we'll be happy to take those questions. All right. If there are no more questions, we will conclude this session, and we'll look forward for you to join the programs, or let other people know who might be interested and who can benefit from this training. Thank you so much for joining today's session, and I hope that it would be great. So there are a few questions. I'll just answer those. So Dhruv is asking for combined costs. So Dhruv, if we have your email address, we can share with you those and provide the scholarship or any discount if we can. So to answer, there's a question from Charles. So I wish to ask for scholarship. Will it be different from the summer training and any other on OmicsLogic platform? So obviously the topics that we discussed today, Charles, and the pedagogy that we discussed today, that would be the path for the session, for the programs and for the training. So I think you can compare to a undergraduate or a postgraduate level course. This will be a little slow paced, and it will be more start from a very beginner level, and then we will move to different levels based on the progress and the time that one allocates. So that would be the major difference, I would say. So thank you, Sparsh, for sharing the scholarship form. And Dhruv, yeah, we can reach out to you. If you have pre-registered, we will have the information to reach out to you. So Sparsh can reach out to you with the cost for the 3 programs and the scholarship or the discount code. All right. So if there are any more questions, I'll be happy to answer those. Okay. Thank you, Charles, for your kind wishes, and thank you everyone for joining today's session. And I hope that we get an opportunity to learn from each other. Thank you so much. Have a good day. Bye-bye.